Started. First of all, let me thank the brave souls who are here, who crossed the frozen tundra to come to this really amazing lecture that we're going to hear today. I also know that there are lots of people online, and I apologize for the late start, but we didn't want people to rush here because it is slippery still on campus. Our speaker today is Sam Mabulaiteye, and Sam is a senior investigator in the Infections and Immunoepidemiology branch in the Division of Cancer Epidemiology and Genetics in NCI. Sam got his medical degree from Makerere University in Kampala, Uganda, and got his PhD in epidemiology and biostatistics at Cambridge University in England. He went back to Uganda and did his further medical training at Makerere University, and then joined the Uganda Cancer Institute and began his studies of interactions of the environment with human disease, studying human immunodeficiency virus, HIV, and the effect of that on cancer in both children and adults. He then moved to the Uganda Virus Research Institute, and following that, he came to the NIH to work as a research fellow in DCEG. Now, in DCEG, his work has taken a few different turns, although he still maintained his interest in the epidemiology of HIV and its effect on human cancer. And he studied two major issues. I think his great love is to study Burkitt Lymphoma and understand the interplay of factors that lead to the high incidence of this disease in children in Africa. And I think he's going to be focusing a little bit on that in his talk. He's also interested in Kaposi sarcoma, which again is an interesting disease in terms of the fact that not everyone with HIV gets this disease. There's obviously a role for herpes virus 8 and other environmental factors. So I think without further ado, Sam, let's begin your talk entitled Burkitt Lymphoma, a Model of Polymicrobial Carcinogenesis and Global Oncology. Yeah.
Thank you, Dr. Gottsman. I would like to express my absolute pleasure for you inviting me here to share about my work. It's really a unique honor for me, and I appreciate the opportunity not only to present my work, but also to receive the feedback that will be coming from all the listeners here, as well as those who are watching the video cast. As you'll see, some of my work will appear preliminary, but that's because the field of Burkitt lymphoma research stagnated after going through 10 years of really vibrant, exciting research. There was stagnation until recently when studies have been initiated to answer outstanding questions. My talk will cover some historical perspectives just to bring everybody to the same page, but I'll mostly touch on malaria and EBV as etiological factors, specifically reviewing the evidence for association, and also describe some of the ideas that I have borrowed from malaria vaccine and diagnostic studies to expand the knowledge of what we have about associations with BL, discuss the potential for proteome and genomics technologies to shed answers to the outstanding questions. And then I hope to just illustrate how BL serves as a model for international as well as multidisciplinary collaboration, as well as some trans-NCI and trans-NIH collaborations. Throughout my talk, I hope you'll really appreciate that BL is a complex model of carcinogenesis, but I would like to first say that I'm very pleased that the NCI has placed priority on Burkitt lymphoma. My invitation to speak here speaks to that. But this point was made amply by Dr. Vamus when he made comments to the participants of the Global Health meeting that was organized by the NCI Center for Global Health in 2012. First, the story begins with Dennis Burkitt, and I'm really very excited to talk about it because Burkitt lymphoma is probably the most amazing story that has come out of Africa. This is Dennis Burkitt. He was an Irish surgeon with one eye, but an extraordinary vision. And part of what I'll describe talk to that extraordinary vision. It was his work in Africa, particularly his ability to convince people to get interested in obscure African tumors, that really unleashed the tidal wave of discoveries that followed in tumor virology, tumor biology, cellular biology, immunology, as well as international collaboration. When you think about Burkitt lymphoma, most people think about a child with a large facial tumor that distorts his appearance. But we do know that Burkitt lymphoma is a B-cell lymphoma that occurs as 3 subtypes: endemic, sporadic, and immunodeficiency associated. We know it's the commonest childhood cancer in Equatorial Africa where malaria occurs. We also know it's a rapidly growing tumor, which suggested right from the beginning that it might have a short latency as well as it could be curable. It is fatal without treatment. However, if you have treatment, it's probably one of those cancers that you'd wish a child gets because the outcome is usually very good. Burkitt lymphoma's seminal Dennis Burkitt's seminal contribution to Burkitt lymphoma really began with that map describing the epidemiology of Burkitt lymphoma in Africa. And what he found was the disease was common in areas where mosquito-borne diseases were common. And that led to the first hypothesis that there might be a viral factor that was being transmitted by mosquitoes that was causing this disease. This attracted interest of Epstein, and Epstein was able to convince the Medical Research Council to give him a small grant for virus discovery in cancer tissues from Africa. And that led to the discovery of Epstein-Barr virus, which we now know is the first human virus to be linked to a cancer, and it's been linked to multiple cancers since as a carcinogen. However, studies quickly showed that EBV was ubiquitous and it was not vector-borne, and that observation led Burkitt and his colleagues who were working in Africa to in fact reformulate their hypothesis that the vector-borne disease was not EBV, but malaria, which was cooperating with EBV to cause Burkitt's. Dennis Burkitt was a surgeon, and I'm pretty sure that he was frustrated that his surgical techniques could not work on Burkitt lymphoma kids. So he was really willing to try anything to help these kids, and he accepted to test chemotherapy and was the first to demonstrate that, in fact, cases could respond dramatically, and some of them were cured when he applied his single chemotherapy. There was a lot of interest in the findings that Dennis Burkitt was describing along with other scientists who are working on this issue. And those findings attracted interest of scientists here at the NCI in the intramural program, which culminated in establishing the Uganda Cancer Center, which was led initially by John Ziegler in 1967 in Kampala. John went to Uganda and shaped this center into a world-class center, began training local doctors to become research scientists, and in fact the work was so robust that many years later, when I joined the medical school, I was also taken up into that field and became a cancer epidemiologist. I had the pleasure of working with John in the 1990s when he came back to Uganda. But there were also interests in the epidemiology program, and here I'm showing work that was done at Colombo University, led by investigators from DCG, Bob Bigger, Paul Levine, and they went to Ghana and established a built epidemiology program, which I have benefited from. I feel very fortunate because I've had the chance to work in the Uganda program as well as the Ghana program. However, let me go back to why I've been very interested in this condition. When I was training as an internal medicine specialist in Uganda, I was very interested in initiating my research career in Burkitt lymphoma. So I went back and reviewed the evidence that we had about the link between Burkitt lymphoma and malaria. And I started with Harrison's Principles of Internal Medicine, and I'm showing here an example from the 15th edition. What I found astonished me. It was just 2 sentences in a chapter of 10 pages in a book of more than 2,000 pages. And basically all it said was it's possible that malaria-related immunosuppression provokes infection with the lymphoma viruses. Burkitt lymphoma is strongly associated with Epstein-Barr virus. The prevalence of this childhood tumor is high in malaria areas in Africa. Just that. For this tumor, which was so amazing that it attracted the NCI to go and establish 2 research centers in Africa. The main limitations of the data that I could find either in Harrison's or in the literature that I reviewed was that there was no information about the direct causation of malaria and Burkitt lymphoma. In particular, there was no information on the magnitude of risk if one was experiencing lots of malaria episodes. There was no information about the specific malaria proteins that were involved in Burkitt lymphoma or EBV proteins, or the role of genetic diversity of the pathogens, EBV or malaria, in influencing the risk for Burkitt lymphoma. And there was no information about genetic risk factors of Burkitt lymphoma. Clearly, when I looked at the evidence closely, I realized that most of the statements being made about the link between malaria and Burkitt lymphoma was based on ecological studies. And I'm showing here one example from 1963, just 5 years after Burkitt described Burkitt lymphoma. And you can see that the distribution of Burkitt lymphoma, which is shown as dots, corresponded to the distribution of malaria in Africa. This evidence has been repeatedly shown in different studies looking at this relationship at different scales. And I'm showing one example from Kenya in 2007, and you can see very clearly that areas where Burkitt lymphoma incidence is high are the same areas where malaria incidence is very high. This type of evidence is limited. It is inadequate for causal inference, and it doesn't tell us any information about the proteins that might be relevant for this relationship. So that's about the time when I met John Ziegler. He was in Uganda doing his study of HIV and cancer in adults, and we decided to add a pediatric component to that study so that we could study, one, the impact of HIV on Burkitt lymphoma and other pediatric cancers, but I was particularly interested in the relationship between malaria and Burkitt lymphoma. John was working with colleagues from Oxford and the CDC, and together we were able to test patients that we enrolled at Mulago Hospital for this whole season extract as a proxy for malaria exposure. This slide shows our results from that study that looked at 126 cases and 70 controls. Basically, on the y-axis, I show the antibody titers to the whole season lysate, and on the x-axis, I show the odds ratios. The one line shows a point of no association, and if the association is on this side, then that's a decreased risk, and if it's on the right side, that is increased risk. So we clearly showed that in these children, having high antibody titers for the whole season lysate was strongly associated with Burkitt lymphoma. My colleagues from Oxford and the CDC went to Malawi and essentially replicated our findings with an observation that kids with Burkitt lymphoma were more likely to have whole season antibodies in their system. These results were quite insightful, but in fact, that was the entire world literature on the association between malaria and Burkitt lymphoma, and it confirmed the link, but it did not answer the questions. For example, one question that wasn't answered was, since we know that being exposed to malaria triggers immune response, resulting in clinical immunity to malaria, and in some cases, parasite immunity, does immunity to clinical malaria protect against BL? That question was not answered by the two studies that I just reviewed. So when I came to the NIH, I started digging into this question. My limitations included the fact that I could not pick from the shelf antigens or candidates that were well accepted by everybody as contributing to malaria protection. If there was a vaccine, I would have just picked the vaccine. But I decided to go through the vaccine candidates, and I picked SE36. I was very interested in that one because there was some evidence from people who were evaluating it as a vaccine candidate that it does block parasite growth in vivo and in vitro. There was evidence from Uganda and elsewhere that showed that people who had high antibodies to SE36 were protected from severe malaria. The studies from Uganda were impressive for me because they were done in an area where malaria prevalence is so high and the mosquito bite rates are so high, the highest that have been recorded in the world, 1,500 bites per year or 5 infectious bites per night. So I thought this was quite impressive. And what really made me focus on this antigen was the idea that the people who were studying it were interested in collaborating with me on my questions in BL. So we put together a panel using data from the NCI Ghana Fukushima project that I spoke about. This biobank has samples from 1965, 1994, old samples, but quite useful for my case. And we had 657 cases that were mostly histologically confirmed and 498 controls that we studied. And the samples were tested by Professor Horai, who studies SE36 in Japan. This slide summarizes our main results. You can see these are the Shizont titers, these are the odds ratios, and these are the 95% confidence intervals. And what we found was, compared to those who were high, kids with the Burkitt lymphoma were more likely to have low antibody titers, showing an inverse association. I was very excited about these results, but people pointed out that you only had one test. You did not have an antibody that is compatible with exposure. So that was a limitation. And therefore, I went back to the drawing board and decided to now pick 2 vaccine antigens. I added MSP1, which was being tested, and then also added 2 diagnostic antigens, HRP2, which is used to measure the burden of malaria or to do routine diagnosis of malaria, and 6-9P, which is used by people who are testing CSP-based vaccines to determine whether somebody has had natural infection or not. This slide summarizes our main results for SE36. I should add that this time I changed my test lab from Japan to George Washington University because I wanted to replicate the SE36 findings in a different lab. So I worked with a colleague at George Washington, Jeffrey Bettany, and basically we showed that kids who had antibodies to SE36 had a lower risk for Burkitt lymphoma, confirming our initial observation. However, what was even more interesting was in this set where we now had data on another antigen, HRP2, we found that kids who had antibodies to this antigen had a higher risk for Burkitt lymphoma. And when we combined the 2 tests and categorized individuals as either not exposed protected or exposed not protected, then that group had 18 times the risk of Burkitt lymphoma compared to those who were in this group. And I began realizing right there and then that we need to use multiple antigens to evaluate malaria exposure. We agree on the association, but the effects depend on which antigens that you are evaluated. We think there are 2 types of responses that are being conveyed. One is a risk response, which is by antigens that elicit antibodies that are irrelevant for protection, and probably protection that is generated by antigens that elicit protective immunity. My only limitation here now was I only had 1 antigen that I had demonstrated protection for. For HRP2, at least I could count the whole Shizont assay as a second antigen. So I decided to select another antigen. And I went to variant 2 CSA. This is an interesting choice because variant 2 CSA has been linked to placental malaria. Placental malaria occurs due to sequestration of malaria parasites in the placenta, and it causes intrauterine growth retardation, abortion, as well as maternal anemia and sometimes death. However, studies have shown very clearly that antibodies to variant 2 CSA block this sequestration process and prevent placental malaria. What was interesting for me, there was literature suggesting that you can actually find antibodies to variant 2 CSA in men as well as in children, suggesting that it's actually there. And I was very interested in finding out whether it might have a link with Burkitt lymphoma. And like for SE36, I decided to collaborate with somebody who studies variant 2 CSA on a daily basis, Professor Ali Salanti in Denmark. This slide summarizes our results from the study that we did with him. We looked at 121 cases from the Ghana Biobank and 101 controls, and we decided to test the samples using 3 technologies: an ELISA-based technology, a flow cytometry-based technology, and a Luminex-based technology. And what we found was that for all the tests, we had exactly the same result, a protective effect, suggesting that these results are likely valid and probably do suggest a mechanism. We know how variant 2 CSA works, but we don't know how it works in the case of BEL. This was a new finding that was totally unexpected for us. We think that maybe BL tumor cells might express CSA. That's what my colleagues suggested to me. And if that was the case, then variant 2 CSA positive parasites would adhere in the tumor, leading to a weak antibody response. We thought that this one is an issue that we could test, and we did that by looking in our tumors. These are representative samples from 40 tumors that we had from our biobank, and we tested them by immunohistochemistry to check to see if variant 2 CSA can be detected in tumor cells. You can see an example of high expression, but it's not in the tumor cells. It is in the stroma. This is low expression, but again in the stroma. So this suggested to us that variant 2 CSA might exist in these kids who have BL, but it's not being produced by the tumor. So the low antibody response is not tumor-induced, but is probably inherent in the innate immune response by these kids. Our follow-up is to test the samples that we have for CSA and probably other glycans, particularly heparan sulfates, to see if they are freely circulating in the blood. And this is really making us think in a totally different way about how malaria might be interacting in these kids to induce Burkitt lymphoma. So I've showed you some results from studies where I kind of cherry-picked antigens. And as I was doing this, I realized that probably that's not the most efficient way to do this. The best way would be to use PF arrays, scan thousands of peptides, and see which ones are interesting in the positive direction and in the negative direction. And what I'm going to do is show you results that I have from working with Denise Doolan at Queensland Medical Research Institute in Australia to use PF arrays to test the feasibility of using them to discover interesting or relevant peptides. This slide summarizes results that we have generated by probing serum of 32 cases and 22 controls. And what you can see very, very clearly, here we have the columns representing subjects, and the rows are representing reactivities for the different antigens. And what you can see very clearly is that our cases and controls are very similar in their reactivity. And this is reassuring because we matched them on area. They also have similar magnitude of antibody responses as well as breadth of antibody responses. These results are important for two reasons. First of all, they show that you can use an array biomarker to confirm similarity of exposure. This is important in case control studies where you are never sure whether you've matched your cases and controls well on a key exposure. For malaria, you could actually use a biomarker like this to confirm that similarity. Secondly, we did an analysis in a restricted set of peptides that were immunodominant, and we found that a small number were significantly associated with Burkitt lymphoma, 15. They had odds ratios ranging from 2 to 7, so suggesting that in fact there might be a way of discovering peptides that are important and relevant to this process. This work is in very early stages, and we are planning to expand it to use the biomarker for matching cases and controls, but also to scan additional cases so that we can generate hypotheses as well as test the possibility of getting a biomarker developed, a risk biomarker for Burkitt lymphoma. So up to this point, my experience studying Burkitt lymphoma have showed me what the real challenges are. I don't think the challenges are those of ideas, what is relevant, what is interesting to study. The challenges have been really about sparse data that's out there to generate new directions because there have been few studies, or the studies that have been there have few subjects that have been included. And in my case, most of the samples that I've been using are rapidly diminishing. The Ghana biobank will soon have no samples left. So if I have an idea, I wouldn't be able to test it. With respect to data, we've had difficulty with imprecise exposure measurement. Which exposure should you use? And also lack of pathology, diagnostic misclassification. All those are inherent challenges that we've had to deal with. So as I was thinking about that, I thought it was time to renew this biobank so that we could answer the questions that we had in mind. And I teamed up with investigators at the NIH. One of them is Dr. Bhatia, who always helps me think through the EBV questions, but as well as the malaria immunology questions. Dr. Gedat, who is here, he's the gentleman who recruited me when I was still in Uganda and invited me to work here at the NIH. I appreciate that. Ruth Pfeiffer, who brings statistical expertise, and Dr. Chanok, who brings all the population genetics expertise that I need. I invited him to join my team before he became my director, but I appreciate the fact that he continues to show an interest. So the study we're doing is being conducted in East Africa in 3 countries. These are the areas where we conducting the study. And basically our plan is to enroll 1,500 BL cases who are aged less than 16 years from defined geographical regions that is histologically confirmed, and we collect clinical and demographical data. And from the same areas, we're interested in getting 3,000 representative matched controls of the same age, same area, but do not have BL. And we collect extensive specimens as well as questionnaire data, blood, saliva, stool, tumor tissue, and questionnaire data on a range of risk factors. Working with this study has given me very interesting insights on what I couldn't do before. And I'm just going to share one example. The question which I had in mind was, if you look at a child who has Burkitt lymphoma, when they've just come to you, are they more or less likely to have current malaria if they live in an area where malaria transmission is so high? In other words, do they have worse or better immunity for malaria than age-matched children? We are able to look at this question because we are measuring malaria positivity in the cases and controls using the thin, the thick, and the thin film. These are examples of positive slides. And we also use the rapid diagnostic test, the HRP-based one, which I mentioned. And here's an example of somebody who has complex infection, 1 and 2 positive strips there. And this is just simple infection, and this is a negative test. So using these 2 tests, you can actually determine whether somebody has current infection or recent infection, because if they have recent infection but not current, they'll be negative on their microscopic result but positive on this one. So you can ask the question that I posed for myself. My results have been very surprising. The first thing that we have found was kids with Burkitt lymphoma are less likely to have current or recent malaria compared to controls who come from the same area. The risk is much less, as you can see there. If you look at the amount of parasites in blood, it's the same thing. You find lower parasitemia rates compared to the controls. The controls are healthy children whom we enroll from their homes. My first thought was that maybe these kids with Burkitt lymphoma have been treated for malaria. So we asked them questions about outpatient treatment for malaria. This is mild treatments. And what we found again was, if you look in the past 12 months, kids with Burkitt lymphoma are less likely to report a history of treatment for malaria as an outpatient. And we also asked them about severe malaria. Are they likely to be treated? And we found again that kids with Burkitt lymphoma are less likely to be treated for severe malaria. And if you put these variables together and we put other variables that we think might contribute to this relationship into the models, and this relationship remains, it seems that the association between current or recent malaria are independent of a history of recent treatment for malaria, either as an inpatient or an outpatient. And therefore, they're not confounded by that, as you can see there. So my results that clinical malaria is lower in BL cases suggest that kids with BL have clinical immunity and probably effective clearance of asexual parasites in the year preceding the time when they get Burkitt lymphoma. This was unexpected because malaria dogma implies that malaria immunosuppression increases EBV load. What we are finding is that, in fact, the kids with Burkitt lymphoma have lower malaria risk. It seems to me that this is probably part of a Darwinian process that we're observing, and I just wanted to lay out the facts. about 200 million cases of malaria occur globally, and about 1 million deaths occur. The numbers are slightly lower for the deaths now. And on about 6,000 BL kids occur, and they all die if you don't treat them. Previously, our thinking was that Burkitt lymphoma mortality arises from this group, which is vulnerable to malaria. My results are suggesting that, in fact, they probably arise from a different group that has resisted or has innate resistance to severe malaria mortality. The mechanism might include the way they handle their glycans in their system. Probably is genetically controlled. But anyhow, I wanted to kind of link some of my findings to this publication. Investigators in DCG in 2013 described a new gene called interferon lambda 4. And this gene has been selected in such a way that people who are in Africa do have the wild type, but people who are in Europe and in Asia have lost the wild type. They have a derived variant. And having the derived variant results gives them an advantage in how they clear hepatitis C virus infection. What really interested me about this gene was the fact that it has very different distribution in Africans and Caucasians, and it has some viral effects. And the people who discovered it, Dr. Rocklina Olson here and Thomas O'Brien, work in DCG. So I wanted to see if there could be some BL effects with this gene. I'm going to summarize very briefly the results that we have on a very small panel of samples. What we found, this is the wild variant, this is the derived one. And when we look at our BL cases, the risk increases with each TT allele you have. In the recessive model, you find that it increases risk, even in the dominant model. It does suggest to us that this could be a genetic factor that contributes, probably through an innate mechanism, to EBV, to BL risk. And Dr. Olson is working with others, including Jeffrey Cohen, to try and find out if there's an EBV angle to this gene that could explain some of these very interesting findings that we are having. I would now like to shift gears very, very briefly and talk about EBV variants. It's been speculated for a very long time that the distribution of Burkitt lymphoma could also be explained by EBV variants that are tumor-specific. This idea was suggested by Kishore Bhatia here, and in 2009 we reviewed the evidence that is there to support it, and we found that in fact there was no clear evidence to support it one way or the other. So we decided that we would look into this question because I had tumors from Ghana, and I was able to get tumors that Kishore had worked on from Brazil. So look at EBV variants in tumor EBV from two different geographical areas. And basically the idea is that you could actually find the sequences that are relevant if they are common in the two geographical areas, but you would be able to also separate them from sequences that are irrelevant but are tracking the geography because they are in that geographic area but they're not relevant for. This slide shows results from our 4 BL tumors from Brazil and 5 tumors from Ghana. And you can see that we're able to sequence full-length virus from the tumors. And you can see that we had about 27 to 90 EBV copies per cell that we found. This work has been done by colleagues at the FDA who have sequence capacity to do this and the bioinformatics to analyze the data. What was very interesting for us was when we did a full genome phylogenetic tree analysis, we found that all the BL cases clustered together, or I should say 8 of 9, because there's one that clustered here, and they were distinct from the NPC that are published in the database, and also different from the health samples that are published in the database. This VGO is from Brazil and clusters with these individuals, so probably it is EBV from somebody from East Asia. We've looked at the clustering at different gene levels, and here I'm showing results for LMP1. And what we found was very interesting. Again, they clustered together the BL tumors from Brazil and Africa. This one remains an outlier, but they're distinct from NPC and distinct from the health samples from individuals from US, Africa, and Brazil. When we look at where the variation occurs, we found that in fact it is limited to just a few genes, about 21 genes. But in fact, those variations are limited to probably 2 genes. These are variations in tumor genes. We are very excited about these results. They are very preliminary, but they do suggest 2 things. One is that the BL-EBVs from 2 continents are distinct and similar in certain respects. They're distinct from NPC, they're distinct from healthy individual, EBV collected from healthy individuals. They have unique variations in EBNA and LMP, unique or common to them. The LMP variations are new, they haven't been described in the literature. The EBNA variations are some of those that have been described in the literature, particularly in tumor viruses from Africa. So to start concluding, I hope that my talk has really shown you how the intramural program has offered me a wonderful opportunity to do my research on BL. The study that I'm doing is an intramural-led study, similar to the studies that were done 30, 40 years ago from the intramural program in terms of starting from the NIH, but different in certain respects because we focus on just malaria, EBV, and genetic risk factors. We think we'll be able to answer those questions. However, the study has also provided a platform for innovative collaboration, and I'll just mention a couple of them. The first one is with Miller, which I mentioned before, but we also have a collaboration with Michael Inard looking at magnesium and EBV in kids who have BL. We are working on a BL molecular staging project with Dr. Warren from the Fred Hutch, and we have a project with Dr. Gurolin Elizabeth from Oslo to look at the epigenetics of Burkitt lymphoma. There have been other trans-NIH collaborations, and here I want to just acknowledge the contribution by Dr. Steve Reynolds, who works for Tom Quinn. He's been very helpful in my work, particularly with logistics, ethics, and shipment of samples, as well as long-term storage in country. I'm hoping that this will expand to other NIAID investigators because they have more expertise in malaria. And then I want to highlight the collaboration we had with Julia Royo when she was at the NLM. That was with health communication. When we started doing our study, we did not have any information about how to talk about Burkitt lymphoma, and NLM helped us with that, linking it to their malaria work as well. And then recently we've initiated a collaboration with Fogarty International Center for principal investigator training, because I have discovered that my colleagues in Africa require some support in terms of principal investigator responsibilities. My work with Fogarty started with Elise McKenzie, and I just want to acknowledge him for telling me that it was not right for me to do Burkitt lymphoma research in Africa without highlighting the terrible mortality for the kids who get Burkitt lymphoma in that region. Diagnosis is a problem. People who work on Burkitt lymphoma know that this is a problem. We are collaborating with CIT engineers, Tom Pohide and John Kareka, to implement CRADA 0270 to develop a benchtop ultrasound-assisted formalin fixation and processing device that could be used in small centers, particularly for hematological samples which don't fit well in the big tissue processors. We think that the work that we're doing, particularly collecting very nice tumor tissues, is going to be very useful for this project, which is led by the Center for Cancer Genomics here at NCI, and might provide an initial entry for African cases in precision medicine. I would like to thank, in a special way, these individuals, Robert Bigger, Joseph Raumeni, and John Ziegler, because they pointed me in this direction and have been very supportive throughout my career. But I also like to thank all the others who have helped me, especially my colleagues at the NIH. You can see my colleagues that I mentioned. Stephen Channock is here. Louis Stout, we've done some work with him. Kishia Bhatia, who is always advising me, and James Gedard, as well as these others. And with that, I'll be very pleased to take some questions.
Questions? And I ask that the people asking questions go to the microphone because there is an audience listening in.
So you showed very nicely some polymorphisms or changes in Epstein-Barr virus nuclear antigen 1, EBNA1, in individuals with Burkitt lymphoma. And as you know, EBNA1 is the major protein that's expressed of all the EBV latency genes in Burkitt lymphoma, and it's a target for cytotoxic T cells. Do you know if the changes you saw in EBNA1 are changes in cytotoxic T cell epitopes, and whether some of these changes you saw in the EBNA1 sequence might correlate with immune escape, so to speak, of Burkitt lymphoma from control by cytotoxic T cells?
No, we haven't done any functional analysis of this data. What I have now is just where the changes are occurring, and what was very exciting for us was to find these changes common in tumors from two different geographic areas. They are all tumor viruses, so they must have the signature that is relevant for carcinogenesis. We plan to do some additional analysis myself, but mostly my colleagues at the FDA, and working with Kishore here, who has a wealth of knowledge about EBV in BL tumors, to try and understand what those changes mean. The way I arrived at this point was I was trying to figure out how do you study the relevant EBV variants in an epidemiologic setting. And there are two possibilities that we're thinking about. One is, let's get EBV from healthy people and find all the variants that are out there, find out how to measure them well, and then do a case control sort of comparison. That approach has not worked very well. We've worked here and we didn't succeed easily. We were working with Paul Ferrer on that. And then we said, why don't we start with the tumors? Because the tumors actually are a sampling of EBV from those populations that have progressed to disease. So they should have the signatures that you want. And if you can narrow down the regions, gene regions that are important, then you can just focus on those regions by using PCR amplification and then NGS sequencing to find the relevant variants. And then those variants that come out in a case control setting are the ones that you'd probably move to study for function. My studies have been limited when I think about function because I've been working, I say working alone as an epidemiologist, as a clinician, but as I've moved out, I've realized, no, you cannot do good work that way. You always end up with half the answer. So I'm very interested in expanding collaboration with people who do bench work, who can knock in, knock out genes and confirm which ones are relevant and which ones are irrelevant and how they are relevant. So maybe we should talk, and we have samples that we can study to clarify those questions.
Thanks.
Steve.
Thank you, Sam. I have a question about the age. What's really striking with the information that you provided about the recent malarial infections, what's really striking is that in this part of the world that it's a very restricted age in which you see the individuals who are hit with Burkitt's lymphoma. You don't see it after adolescence per se, and it's not very often even seen people making it into adolescence. So in that regard, what are your thoughts about the age as a reflection of developmental opportunities or developmental, periods of time in which someone would be at risk for developing this? Have you started to look at, or think about the question of the burden of malaria over time, that may over time just help to get someone to an immunologic state where they aren't necessarily at risk for this? Or, it's such a curious thing that it occurs in such a small age range, when in fact malaria, affects 400, 500 million people across that belt. What are your thoughts?
Right. So we are thinking about that. Our thoughts began with collecting good data and samples. So we asked many questions about fevers. So I just presented the fevers that are malaria, but We also ask questions about fevers that are not malaria, and based on the report from the patient's caregiver. So we're able to really say that these findings are very specific to malaria. They are not there for the non-malaria fevers. We also have samples. We have blood clots, so we'll be able to look in that to quantify the malaria experience. But you are correct that it's striking. If malaria contributes, and we know that malaria doesn't go away in that region, people get bitten by mosquitoes every day. Why is it that the risk for Burkitt lymphoma peaks? It does suggest that there is either developmental process that cuts down, the risk process, and it might be driven by malaria immunity, or it might be driven by B-cell physiology, and I'm not sure which is which. Our kids are matched nicely for age, so we know that the results that we're finding in the cases should be similar to those in the controls because they're all about the same age. They're all 7.4, 7.5 years, so they're old kids. In these old kids, the cases have better immunity to malaria than the controls. So previously when I was thinking about how to design a study, I would have focused on the kids who are vulnerable to malaria as the kids where you are likely to find the translocations, to study the longevity of translocations if you could do that. Right now, I think, no, you must study the kids who are hardy, those who are able to control the malaria infection. From the epidemiology perspective, if we can have a biomarker that allows us to separate these kids into phenotypes, the phenotypes that we've been thinking of are, there's a phenotype that leads you to severe malaria, and your risk for death is very high if you're in that phenotype. There is a phenotype that leads you to asymptomatic malaria, and your risk for death is low in that phenotype. But your risk for Burkitt lymphoma might be higher because you don't have overt clinical malaria, but you do have submicroscopic malaria. That might still be driving the process. I hope that with our willingness to collaborate, with different investigators who bring different strengths to the team, we should be able to generate the next steps, to answer the question that you asked.
Thanks for an interesting talk. You mentioned you studied other fevers. Did you or anybody else find any association between the incidence or severity of malaria and the incidence or severity of other viral diseases such as chickenpox?
No. So our samples are cross-sectional, and the questionnaire allows us to look back in the history of the kids' experiences. We did not ask specifically about chickenpox. Sorry, not chickenpox. But we can't answer that question because we just used cross-sectional data. In the samples that we have, you could do a study by measuring for specific infections and show if there's a correlation between malaria, for example, and those infections. You could also use the data that we have on the history of malaria experience and see if there's a correlation. What I have done, and this was just to ask, if you've had an experience of severe malaria in the past, let's say 1 year, are you different in terms of your current prevalence for malaria now than somebody who reports they've never had? And we found differences. We do find that kids who report that they've had a previous episode of severe malaria are more likely to have current parastemia than those who don't. And that's striking because we're talking about healthy children. I mean, not healthy but in the African context, they're in their homes playing and doing their stuff. So they're not sick, and they have parastemia. So it does seem like having severe malaria is a different phenotype from never having an episode of severe malaria. It has to be immunologically driven, and I'm not sure exactly what the immunological processes are, but they're there. I have had some discussions with Sue Pierce and Louis Miller, as well as Patrick Duffy, And their focus has always been on severe malaria. But it seems like we must also focus on kids who reside in areas where malaria is so prevalent, but never seem to suffer from it.
So Sam, thank you very much for a very insightful talk. So the prevailing dogma, the work may well have been inspired by that, and that is geographic clustering of EBV and malaria parasite incidents. But your surprising findings of a relationship almost also underscores complex pathophysiology, and you hinted at the glycans, parasite sequestration in placenta, and of course, the correlates of that may well be at a molecular level. When you look at these kids, do you also find that there might be clustering at the level of other co-endemic parasites that could modify disease incidence vis-a-vis the immunologic paradigms that you are beginning to hint at in your answer to one of the questions? So I'm quite interested in what you think may well be going on. So if you subtract malaria only for a moment and consider the other parasites in those geographical areas, whether you think you might arrive at correlates that might be even more informative in terms of what's really going on at a pathophysiological level. Thank you.
So we take stool samples and measure for stool parasites. We've not found appreciable levels of infestation for stool parasites to make any meaningful contribution to what we're seeing. We're very surprised about that. But then I realized that the immunization program, expanded program of immunization, they give out the bendazole or albendazole as part of the visits that the kids, the treatment that the kids get when they visit. So that has reduced parasite load. Maybe they do have lower levels. So we are not able to look at that using the data that we already have. If we can find a good serological test, we might look at some of the parasites to see if they do contribute at that level. The ones that we've looked at are EBV. We've measured EBV in some of the cases, and I didn't present those data. We do not find asymptomatic malaria to influence EBV levels. So the kids who are EBV, who are malaria positive and malaria negative, don't have appreciably different EBV levels. But this is because we are looking at asymptomatic kids and not kids who have severe or acute clinical malaria. Those tend to have different EBV levels. We're very interested in interaction with other pathogens. KSHV is one that I'm very interested in because it occurs there. It influences the risk for Kaposi sarcoma. I'm interested in seeing how it interacts with malaria. There's some studies that are showing that there is an interaction between KSHV and malaria. So we'll need to see how KSHV, EBV, parasites, and maybe other pathogens, could contribute to this whole interaction to either increase or decrease risk of disease.
Great talk, Sam. It just came to mind. Do you know that the BL tumors that you sequence the EBV genome, did you look at the other genetic lesions that are also common in Burkitt lymphoma, like the c-myc, and were they all the same, or was there any differences there?
We haven't done those analyses yet, but we have the data. This is a project that is likely to grow. Ultimately, we would like to include the emblem data in this project because we have much more granular data on emblem than we have on the Ghana cases, for example. We don't have detailed questionnaire data. We don't have current malaria data on the Ghana cases. So emblem is going to allow us to ask some of those questions that you are alluding to.
Sam, I have one question. You gave us the somewhat unexpected result that in children who get Burkitt's lymphoma, there seems to be a decreased evidence of recent or recurrent infection with malaria. You didn't expect that result.
No.
And you're pursuing what it seems like a very reasonable approach, which is to look for genetic factors in the children that may account for their decreased incidence of malaria and so on. I guess another formal possibility is that acute malarial infection protects against Burkitt's lymphoma. And one way you could check that as a control would be to look at other genetic factors that are known to protect against malaria, like sickle cell trait. So is there any association between Burkitt's lymphoma and sickle cell trait?
Yes, so we've looked in our samples. Miller here was able to test our samples for the sickle cell gene, and we find a non-significant protective effect. In fact, in the model for IFNAL4, the sickle cell trait does not confound that association, and itself is not significant. So that's surprising to me. We're still exploring how these two fall together, sickle cell, IFNAL4, and probably other genes that we'll be looking at. But what it really told me was I won't be able to get to the bottom of this question if I work alone. So I need to work with other people who bring different...
So that there are beginning to be other genetic markers that people have looked at that are protective. I mean, malaria obviously is an enormously selective circumstance in African individuals. And so genetics probably could give you some hints as to what actually is going on.
Yeah.
Any other questions? If not, let's thank Sam for a terrific talk. And I wish you a very careful walk back across the campus.